Be seated. Let's get started. My name is Xianqing Yang from Agriculture Agri-Food Canada. I'll be chairing this session. As you can tell, my co-chair is not here yet, but it's 5:03. It's a long day, a packed program, lots of content. I can understand we're all tired, but thank you so much for coming to the session. And first, I would like to invite Dr. Ilario Forcino, sorry for my butchering of your name, from University of Torino, Italy. And his presentation will be on impact of a multi-strain probiotic on microbiome of free-range chickens. Welcome.
Thank you so much for the introduction, and thanks for all of you that are here and not around the city to visit this beautiful city. So I hope that you enjoy my presentation about the impact of a multi-strain probiotic on the microbiome of free-range chickens. New feeding strategy is the new research area. Several research groups try to understand how the addition of innovative feed ingredients can help the growth, promote the growth of the birds, reduce the mortality rate, and in particularly what it was possible to observe that several research groups, as we did it in the last couple of years, try to use novel, for example, protein as a new source of protein from insect in order to promote the healthy status of the broiler. Then there are several reports that try to use, for example, prebiotics, in particularly fruit oligosaccharides, in order to increase the resistance of the birds to the external perturbation, and then of course the application of probiotics in order to reduce the incidence of the pathogenic infection in the broilers. In particularly, all these kind of new feeding ingredients help to maintain the healthy status of the broiler from one side, confer resistance to pathogens, but then also can have a beneficial effect on the microbiome of the gastrointestinal tract. On the other hand, the application of antibiotics during farming, from one side, reduce the incidence of the pathogenic infection in broilers, but from the other side, reduce the diversity and the function of the gut microbiome in the gastrointestinal tract, but also can be also an issue because that's an increase in the resistome of the broilers with all the consequences, like, for example, the spread of the antibiotic resistance genes in the environment with all the consequences related to human health. The application of probiotics here in the slide, you can see several beneficial effects of the probiotics that explain when they are using in broilers. But we don't have to forget the synergic effect of the probiotics plus prebiotics, because this synergic effect has a beneficial effect during the growth of the broilers. So aim of the study was to try to see the positive effect of a multi-strain probiotic formulation made by the application of Lactiplantibacillus plantarum and Lactiplantibacillus pentosus from the Turin culture collection, daily administered to broilers. And then the experimental plan. So we start with 160 one-day male broiler divided in 4 different dietary treatments, and each treatment consists in 5 pens with 8 birds per pen. And we have 4 different dietary treatments. The first one, it was just the administration of the multi-strain probiotic daily with water in order to reach the final concentration of 10 to 6, 10 to 7 CFU per broiler based on the daily intake of water. Then we have the second dietary treatment called mix, because it was a mixture of the multi-strain probiotic plus the addition of the 10% in the standard diet of inulin. We choose this kind of concentration because it was reported in literature that this kind of concentration promote the growth of the broiler, while highest concentration of inulin have not a good effect on the broiler because can increase also the inflammation. So we choose this concentration as a balanced one. Then we have one dietary treatment only with the 10% of inulin, and then some broiler used as a control. The experiment lasted 33 days and was performed in the animal farming of the University of Turin. So during the trial, we recorded the growth performance of the broilers. We also calculate also the mortality rate, food damage. Then along the trial, we also collect fecal samples in order to monitoring the gut microbiota composition along the trial. And here we perform the 16S amplicon-based sequencing. Then at the end of the trial, during the slaughtering procedure, we collect also blood samples in order to analyze blood parameters. We also collect organs and gut segments in order to perform histomorphological investigation, and then we collect also cecal samples in order to study the short-chain fatty acid fraction of the cecal samples, and then we perform shotgun DNA sequencing on the cecal samples. Moving to the results, we observed that the dietary treatment did not affect the blood measurements, growth performance, and the severity of histopathological alteration in any gut segments, in any organs that we analyze. This is a positive effect because we can say that our dietary treatment cannot modify the normal healthy of the broilers. And as a positive effect, we observed that the jejunum of the broilers fed exclusively with the mixture of the probiotic display longer villi than broilers fed with only inulins and broilers fed with a mixture of probiotics and prebiotics. Then moving to the microbiota characterization, so we follow the microbiota composition of the fecal samples along the experimental trial, but we didn't see any significant difference due to the dietary treatment, since only the sampling time was the main factors that was affect the microbiota composition. This is in line with literature because during the growth of the broilers there are some physiological change that modify naturally the microbiota composition. But then when we analyze the microbiome composition by doing shotgun sequencing on the cecal samples, we observe, for example, as a confirmation that the highest abundance of the Lactobacillus and in particularly the presence of Lactobacillus pentosus in broilers fed with the multistrain probiotic, and then surprisingly we observe the highest predominance in the cecal samples of Faecalibacterium, mainly associated with the dietary treatment based on the application of the multistrain probiotic plus the inulin. And in particular, we can hypothesize that the degradation of the inulin due to the multistrain probiotic release free fructose, lactate, and acetate that can stimulate the growth, the overgrowth of Faecalibacterium in this dietary treatment. Then when we went to analyze also the short-chain fatty acid composition, we also observed something very interesting. If you can see from the graph, it was possible to observe that broilers fed with a mixture, so with the multistrain probiotic plus the inulin, display the lowest presence of most of the short-chain fatty acid. This is due to the microbial cross-feeding phenomena, since it was reported that in particular Faecalibacterium use short-chain fatty acid as a source of energy. So in this slide we can see that the highest predominance of Faecalibacterium was also related with the lowest amount of short-chain fatty acid that we detect in the cecal samples. And then we observe the highest abundance of butanoic acid only in broilers fed with the multistrain probiotic. This is something expected because we choose the multistrain probiotic for the ability to produce butanoic acid. Then, by using the shotgun data, we try to reconstruct also some genome in our cecal samples, and then based on the previously results, we try to understand if the dietary treatment was affecting the strain diversity, in particular related to Faecalibacterium. And then in this slide, we can see that we reconstruct the Faecalibacterium pangenome from the shotgun data. We obtain several strains of Faecalibacterium prausnitzii. And if you can see here in the phylogenetic tree, we observe that the strains belonging to Faecalibacterium prausnitzii from broiler fed with a mixture probiotic and prebiotic, and the Faecalibacterium that were present in the broilers fed only with the multi-strain probiotics, were clustered together and were very different between the Faecalibacterium prausnitzii strains that we reconstruct from control samples or inulin-fed broilers samples. And in particular, by analyze the function, the pangenome of the Faecalibacterium prausnitzii of mixed broiler, we observe, for example, the highest predominance of genes involved in the metabolism of branch chain amino acids. That can be a positive effect, since the increased level in branch chain amino acid increase the resistance to births to external factors. Then we also analyzed the total microbiome function of the broilers, and in this slide you can see the differences between the potential function of the microbiome of the control birds compared to mixed birds. And in particularly, mixed birds display highest KEG genes for the biosynthesis of lysine, that it was reported to reduce the amount of fat in the carcasses and promote muscle mass, so it's a positive effect during the growth of the broiler. And then we also observe the highest abundance of alkaline phosphatase, that it was reported to decrease the lipopolysaccharide production in the gastrointestinal tract, reduce gut permeability, and then reduce metabolic endotoxemia and inflammation. Then we also evaluate the resistome of the broiler due to our dietary treatments. And in particular, we observe a reduction in the incidence of several antimicrobial resistance genes in broiler fed with the multistrain probiotic plus inulin. And in particular, we observe a reduction in the genes belonging to the class of beta-lactamase, erythromycin, and tetracycline. And in particular, by analyzing also the pangenome of our Faecalibacterium prausnitzii strains, we also observe that the Faecalibacterium prausnitzii strains from control samples display highest predominance of multi-drug resistance genes, compared with the Faecalibacterium prausnitzii strains reconstruct from the mixed dietary treatment and from the only application of the multistrain probiotic. So in conclusion, by our experiment, we observed that our dietary inclusion did not influence bird performance. That can be a positive effect, because we can say, okay, we can apply this kind of treatment without any such of problem during the growth of the broiler. In addition, we didn't observe histopathological alteration in any of the organs and gut segment that we analyze, since it was reported, for example, that the highest amount of short-chain fatty acid or even the inulin in some cases can increase the inflammation in the broiler, but in our cases the broiler were fine. And then we also observed that several KEG genes with potential beneficial effect were observed in the dietary treatment based with the inulin plus the multistrain probiotics. And then in the end, we also observed that our dietary strategy also promote the microbiome selection also at strain level, because we observe a very different composition at strain level, in particularly regarding Faecalibacterium prausnitzii. And then at the end, we also observe a reduction in the prevalence of several antimicrobial resistance genes. And all these findings should be considered in the perspective of developing novel personalized strategy to promote feeding solution for poultry industries. I would like to thank all my colleagues for helping with the big experiment, and especially Professor Luca Kokolin, the coordinator of the IT Food in Poultry project. Thanks.
Thank you very much for the very interesting presentation. We have a question on the web. In the resistance profile, what does the value scale represents?
Okay. The value scale is the correlation between the dietary treatment and the abundance of the antimicrobial resistance genes.
Thank you. We have time for one quick question. One more quick question from the audience, if there is any. Yeah, there.
Hello, this is Petra Zegers. Thank you for a very nice presentation. Would it be possible to sell such products, a probiotic, and sell it to chicken farms? Would it be very quickly possible or not?
Yes, can be. Actually, we did in an experimental farm, so our experimental farm in the university is quite smaller one, but the application of the dietary treatment can be easily scaled up. That's no problem. We also try to also lyophilize also the strains, and they were fine, so we are ready to scale up it.
And it would also be allowed?
Yeah.
All right. Thank you, Dr. Ferracino, for
Thank you.
your very interesting presentation. Our next presentation is through Zoom, I believe.
Good afternoon. Can you hear me?
Yes, we can hear you well. The next presentation is by Dr. Lorenzo Nissen from the University of Bologna, Italy. And his presentation is protein enrichment of gluten-free bakery products by Arthrospera, plantanesis, in vitro study of the effect of formulation and sourdough process on colon microbiota through MIC cold gut model. Listen?
Yes, okay, thank you. Sorry for the long title. And thank you for having me. Thank you for selection. I've been selected. And it's an honor to take part again to the Food Micro Congress. Today I'm presenting this communication that is the recipient of work of preliminary result of about a work conducted and coordinated by Professor Alessandra Bordoni and Professor Andrea Gianotti. And is about the study of the in vitro impact on the colon microbiota of gluten-free bread that was enriched with Arthrospira platensis, that is also known and better known as spirulina. So the rationale was based on a 2021 previous work where we start investigating several formulation of gluten-free doughs and breads produced with sourdough technology or not, and added with spirulina as a protein source, because you know that one of the food technological challenges for the gluten-free sector market is enriching the product with protein, because them are low in its content. And on this preliminary, on the previous work, we found out that spirulina breads after sourdough fermentation can enhance the production of postbiotics, and even there were the production of exclusive bioactives like thymol, borneol. They were then correlated with the microflora of the ferment of the products of the sourdough process, and correlated so to the probiotic potential of spirulina. So we want to go deep, and the next step will be an in vitro testing on a gut model to see if the breads, spirulina breads and the sourdough breads still maintain their bioactive potential. And what we did, we coupled two gut models. We coupled two systems to prepare gut models. When we firstly digested with the InfoJet system that is very well known, the gluten-free breads with and without spirulina in fortification, and with or without the sourdough process. The traditional process was just then made by baker's yeast and fermented just for 6 hours. Then, after digestion, we perform the microbiological part, that is the fermentation on a column model. And then we used to analyze the changes of the volatilome by anomics approaches. So getting to the microbiological setting of the part, the column model was a batch short-term proximal column that was built up by 5 independent bioreactors, which was harboring 0.1% of the digest states of the algae breads and the control breads. Moreover, we have a blank control where the colonic fermentation was underwent with the digestive enzymes. We then perform changes in respect to the baseline, when the baseline is the adaptation time that the microbiota, fecal microbiota, gets to adapt to the colon, to the bioreactor condition. This MICODE have been previously, and the protocols have been previously and recently published, and the analyte values are presented at omics approaches. So we analyze microbiomics by 16S-MISEC and qPCR to be more versatile. And we perform metabolomics analysis through SPMG-CMS. And then we combine the data set performing a correlation, normal Spearman correlation as an interomic showcase. So this is the normal pipeline that we use to produce when we are working with the MICODE. We have even some several quality controls and a robust statistical model. So getting to the presentation of today, the results that I want to present you are preliminary, are those just relative to microbiomics, so the 16S-MISEC, then followed by the anosism, and presented as log2 fold change, and represented by volcano plots. And then we have results about metabolomics that are about SPMA, ANOVA, and multivariate analysis. Getting to the microbiota, we see the modulation that this sample, the control bread traditionally made, the digestates of this bread, once were fermented by the colon microbiota, the family level shown important overrepresentation of Enterobacteriaceae and underrepresentation of Clostridia. And you can see that here on these volcano plots about the species level, we have the red dots that are the overrepresented taxa and the blue dots that are the underrepresented taxa. Those that appear blue colored are just those significant. And we see that there is a confirmation of the Enterobacteriaceae overexpression, overrepresentation with an overrepresentation of E. coli. And underrepresentation also of Clostridiales, sorry. This scenario changed a lot when we ferment the digested sourdough bread. And we have that Clostridiales, the taxa of Clostridiales, have several taxa about at the species level that were reducing. Unfortunately, even some friendly bacteria like the Faecalibacterium that already have introduced Professor Ferrucino. And but there are even important overrepresentation of lactobacilli. And this is what happened when we test the algae bread in a traditional process. We see that these, at the family level, there are important overrepresentation of Enterobacteriaceae and Clostridiaceae. That is not a very positive fact. But there is even, at the species level, an overrepresentation of Lactobacillus gasseri, Enterococcus durans, and clinically debated Clostridium butyricum. But over, unfortunately, a reduction of commensal Akkermansia mucinifila. But let's see what happens when the sourdough algae bread is applied. Still, the sourdough gave a changes in biodiversity more important than the traditional made breads. So at species level, there are important overrepresentation of bifidum, Bifidobacterium bifidum, Lactobacillus gasseri, Enterococcus durans, and even postbiotic producer Akkermansia mucinifila. Also, and very important, there are reduction and the representation of opportunistic taxa like Blautia, Ruminococcus torques, Ruminococcus navus, and the Alister invisus, for example. But unfortunately, a reduction on commensal bacteroides. Getting to the volatilome analysis, I hope that here on these PCA plots you can easily follow what will communicate. But on the left side you have the plots of cases, and on the right side you have the plot of variables. We friendly use this kind of representation because the values are spread on a plot, on a plane. So you see that BL, that is the baseline with the red flags, is discriminated from the fermentation sample. And very important, is discriminated from the, and very important in this plot is discriminated the algae to the control. So we can see on the variables plots that the algae are described by alcohols derived from the matrix, as including geraniol and olefins and other olefins, while the control is described mainly by alcohols produced by colonic fermentation. These highlighted Vox variables are highlighted because are MANOVA significant. The MANOVA which we performed was made categorically made for the matrix and for the time of fermentation. Similar scenario was with higher molecular alcohols, and we can see still that on the left side, the variables that describe the algae type bread, and mainly the sourdough algae type bread fermented at the end point, were described by more antioxidant phenolics and metabolites of phycocyanin, that is an important antioxidant compounds of algae. While that of the control were normal colonic fermentation or also food contaminants. Getting to the aldehydes, we have to point a caution because the algae type breads were described by two nonenal. That is reported to be an oxidation product of several types of fish oils. And then getting to the alkenes that are important, where this class is recipient of very important bioactives, we can still see a strong discrimination in respect to the baseline of fermentation and from the algae type to the control type. And we can see that the algae type at the pinpoint 1 is described by bioactives and antioxidant terpenes, like naphthalene octahedro and MC9. So the discussion of results are that the modulation of the microbiota by algae bread seems dependent on the technological process. In fact, minor effects were seen after colonic fermentation of traditional breads, while some prebiotic features were coming to appear when the colonic fermentation was conducted on the gestate of sourdough bread. But we have even to say that the spirulina sourdough foster more beneficial microbes than the control. Also, getting to the metabolites, the point of caution is related to the oxidative aldehydes that are described in the algae breads, because non-enal or other compounds like that are undesired oxidation compounds of omega-3 and omega-6 lipids, in which the algae oil is rich. And even after a positive outcome on metabolites, is that after in vitro digestion, colonic fermentation, the different bioactive compounds are still present and retained, and so or slightly microbially transformed in order to be more bioaccessible for the host. So the take-home message are that the impact of gluten-free breads on colon ecology of not celiac origin is not prebiotic, but is inducer of overgrowth of Enterobacteriaceae, some opportunistic bugs. But this effect could be mitigated by the process technology. Sourdough fermentation give a positive modulation of the colon microbiota. And lastly, when we combine the sourdough fermentation and their fortification with spirulina flours, we can exert a prebiotic, we can see a prebiotic effect where there were more beneficial microbes and limited opportunistic ones. So the perspective are next, of course, we have to hand short chain, medium chain, fatty acid analysis. We have to hand some other analysis on our pipeline. And even we have to find a correlation between microbiomics and metabolomics dataset. But in this, we can even be advantaged by a machine learning approach that we have recently, a Keyclicks multiomics framework that we have recently generated in our research team. And then to confirm and extend these results, experimentation of in vitro celiac model and maybe even long-term in vitro model will be necessary. But these data could serve as a foundation for clinical application on future nutritional interventional trial, even in order to reduce animal testing following the principle of One Health. And I give even a call to other researchers of these topics in order to find a way to share data set of in vitro gut models in order to harmonize the different gut models that are present and are available for research. Thank you so much, and sorry if I took too much time.
Thank you very much, Dr. Nielsen, for the excellent presentation. We have time for one quick question, if there is any from the audience. Yes, Luca. Yeah, she's coming.
So, hi, Lorenzo. This is Luca from Turin. Nice to see you. I have a very quick question. How was the bread organoleptically?
Thank you, Professor, for your question. And on that paper that was published on Food and Function, we even aside and present the result of sensorial, of a consumer test. And on that paper, even we try to make some correlation with the sensorial attributes. So far, the control bread, as I remember, was better appreciated than the spirulina one. But we can work better too. But if you see, even on the formulation that we prepare, the formulation is very plain. So we just add salt, the flours, and some cellulose, but no other additives. So maybe this feature could be masked somewhere.
Thank you.
Thank you so much.
Thank you, Lorenzo. Thanks again for the presentation. And Now we move on to the next presentation. Our next presenter is Ms. Steliani Dimitra Pipe-Giannelli. Her presentation is antimicrobial resistance of E. coli at single cell level. Welcome.
My name is Stella Papagiannelli, and I am a PhD candidate at Aristotle University of Thessaloniki. And today I'll be presenting to you part of my PhD thesis titled Antimicrobial Resistance of Escherichia coli at Single Cell Level. Antimicrobials are substances used to prevent microorganisms or to stop them from growing and multiplying. And they are commonly used and widely used in human and veterinary medicine in order to treat various infectious diseases. One of the primary tools for assessing resistance levels is the use of the minimum inhibitory concentration tests, which expose bacterial isolates to a series of concentrations of an antimicrobial agent in order to identify the lowest concentration that effectively prevents bacterial growth. Minimum inhibitory concentrations can be determined through a variety of methods, such as the micro or micro dilution method, gradient strips, or diffusion discs. Regarding the broth microdilution method, which is widely used, wells in a microtiter plate are filled with Mueller-Hinton broth that contains a series of concentrations of an antimicrobial agent on a twofold scale. And these are then inoculated with bacteria and incubated overnight. Afterwards, the lowest concentration where inhibition of bacterial growth is present is reported as the MIC for that specific isolate and the respective antimicrobial compound. Afterwards, the reported MIC value must be compared with MIC clinical breakpoints in order to determine whether this strain is susceptible or resistant to that antibiotic. These breakpoints are established by organizations such as the Committee on Antimicrobial Susceptibility Testing, UCAST, or the Clinical and Laboratory Standards Institute, CLSI. An example of a UCAST clinical breakpoint table is depicted on the slide. And in particular, the antimicrobial and the microorganism in concern are gentamicin and enterobacteria, respectively. Gentamicin belongs to the class of medicine known as aminoglycosides, and it works by preventing protein synthesis. Due to the widespread and often inappropriate use of antimicrobials, antimicrobial resistance poses an emerging threat both to food safety and human health, with the World Health Organization deeming it as one of the greatest health threats over time. So let me stress out at this point that the majority of antimicrobial resistance studies, as well as MIC determination studies, are conducted at the population level without and by considering bacterial populations as a whole. However, single cells of isogenic populations can exhibit behavioral heterogeneity of phenotypic origin under various environmental conditions. So, single-cell behavior at the presence of antibiotics can be considered heterogeneous. So the question that rose was if that single-cell variability can affect the output of the broth microdilution method. So, the objective of the present study was to assess the single-cell behavior at the presence of antibiotics and also the implications for population growth dynamics. In order to do so, we utilized a time-lapse microscopy method for monitoring the colonial growth of single cells. The quality of the images obtained was improved by developing an autofocus procedure. Its final image was the result of multiple images taken at different z-axis planes. The combination of the z-stack led to a single in-focus image. The bacterial strain used was Escherichia coli. After inoculation on Müller-Hinton agar that contained a series of concentrations of gentamicin up to 3 micrograms per ml, a small part of the agar was removed and carefully placed for microscopic observation. Images of the selected field of view were acquired every 5 minutes for 24 hours. And the output of the method is a video showing the behavior of the same cells over time. The method allows for observing single-cell responses, the measurement of division times, and also the determination of population dynamics. In our study, the majority of the single cells tested were able to divide and form microcolonies. However, there were a few cells that were unable to do so. In addition, it became evident that the concentration of gentamicin affected the probability of growth of single cells. Characteristically, as the antibiotic concentration increased, there was a decrease in the probability of growth of single cells. We were also able to monitor the division times of the single cells up to 6 generations, and we came to the conclusion that the presence of gentamicin did not affect the division times of the single cells. Also, the high quality of the images obtained from the microscopic method allowed us to count the cells visually. So each image of the video that corresponds to a time point was translated to cell number. By doing so, we realized that gentamicin concentration highly affected the maximum number of cells that constitute a microcolony. Characteristically, as the antibiotic concentration increased, the number of cells in a microcolony was fewer. For example, at an antibiotic concentration of 0.5, you can see that the antibiotic effect is negligible. But on the other hand, at a concentration of 1.75, each microcolony approximately consists of 6 cells. The mean of the maximum population in each microcolony at all gentamicin concentrations tested was further translated to cell generations, which was fewer as the antibiotic concentration increased. So, through our microscopic observations and their further analysis, it became evident that, indeed, single-cell behavior at the presence of antibiotics is heterogeneous. So, we wanted to see if that can affect the output of the broth microdillusion method, which, let me remind you, is conducted at high population levels that do not take into account single-cell behavior. So, in order to do this, we utilized the bioscreen-C method. We used Escherichia coli inoculum levels ranging from 10 to the power of 4 up to the 10 to the power of 7 CFU per ml, and the medium was Müller-Hinton broth containing a series of gentamicin concentrations. So, through the method was able to detect growth when the optical density in the wells in the microtiter plate reached a value of approximately 0.2. And this optical density value can be translated to approximately 7 logs CFU per ml in bacterial population. And let's call this end detection. So, end detection for all gentamicin concentrations tested was not statistically significant. And in addition, end detection variability was further characterized using appropriate probability distributions, and the best fitted distribution was found to be PERT. So, through our findings, coupled with Monte Carlo simulation, it became evident that the observed growth through the broth microdillusion method is of probabilistic nature. For example, if we have an initial population of 7 logs, if we repeat the experiment many times, 90% of the times we'll have bacterial growth and the Zetamis in concentration of 2, and 10% of the times we will not. So this could be attributed to the initial inoculum level, as an increase in inoculum level resulted in an increase in the probability of growth. And also it can be attributed to the variability in the maximum population of microcolonies originating from a single cell, and also the end detection. So if we translate this based on the methodology we use for MIC assessment, we can see that, for example, at an initial population of 5 logs, there is a possibility of 1 that the MIC would be at an antibiotic concentration of 1 microgram per ml. On the other hand, if we start with a population of 6 logs, we can see that there is a 0.532 probability that the MIC would be at a concentration of 1, and a 0.468 probability that the MIC would be at 2. So it became evident that MIC determination is also of probabilistic nature. These findings, our findings were further validated at the population level, and indeed with an initial population of approximately 5 logs, the MIC is determined at a concentration of 1. With an initial population of 6, the lowest concentration that was able to prevent bacterial growth was 2 micrograms per ml, and respectively, at an initial population of approximately 7 logs, the MIC value was determined at 4 micrograms per ml concentration. To conclude, the findings of our study can highlight the resultant of the single-cell behavior under exposure to various antibiotic concentrations, and provide a new perspective into MIC assessment methodologies. And this can be achieved through a probabilistic approach. More specifically, the presence of gentamicin did not seem to affect the division times of single cells, but it did, however, affect, on the other hand, the maximum population of microcolonies originating from a single cell. So there arose a need for a better definition of the MIC that would take into account the individual cell heterogeneity. So I will conclude this presentation and leave you with a question and future challenge that results from our presentation. So taking into account that the high variability that we encountered in the maximum population of microcolonies, it would be of great importance to see the impact of the number of generations on the antimicrobial resistance, on the development of antimicrobial resistance. Well, that's something we have to investigate and present in the future study. Thank you very much.
Thank you. We have time for one quick question from the audience, if there's any. Yes.
I enjoyed very much the connection between the population and the probabilistic symbol. Can you hear me? Yeah.
Yes.
So, not surprisingly, because with your boss, we have been collaborating in this for many years. So I enjoy very much the effort to try to connect the population level dynamics and the single cell. And then I just draw your attention, an interesting fact, it's a mathematical thing, that when we write up the exponential model, saying dx over dt, where x is the concentration of bacteria, dx over dt, and then we say equal to a certain mu times dt, so there the dx over x is the probability. Which means that the dx over x is that the change, relative change, how many are born relative to how many were. That's actually your probability. But then at high level, it becomes the natural log rate. So I'm not surprised that there must be some sort of connection between the two. But this needs to be explored, especially with the MIC value, because the MIC value, as you pointed out very well, the MIC value is in fact becomes a probabilistic value at low concentrations. Yeah. So I really enjoy that. I just wanted to put it in a mathematical way, that when people speak about MIC value as a deterministic parameter, in fact, at single cell level, it has to be probabilistic, actually characterized by distribution. So thank you very much.
Thank you as well for your kind words.
Thanks again for the presentation. Our next presenter is Ms. Natalia Anrath. Her presentation is characterization of a large group of Listeria monocytogenes using whole genome sequencing, assessing their virulence and persistence potential.
Thank you. Hello, everyone. I'm Natalia, and I'm a PhD candidate at University College Dublin under the supervision of Professor Seamus Fanning. And my research focuses on Listeria monocytogenes. So just an overview of my talk. I will give you a bit of a background on Listeria, its significance, virulence, and persistence in food production. I'll talk about the assessment of Listeria monocytogenes using bioinformatic analysis. I'll talk a little bit about my own study then and the characterization of Listeria mono isolates in Irish food production. And I'll have some concluding remarks at the end. So the genus Listeria, Listeria monocytogenes divides across 4 evolutionary lineages, and they can be further subdivided into 14 serotypes. Serotypes 1 to A, 1 to B, and 4B, which are all of lineage 1, cause 95% of human listeriosis cases. And then these can be further divided into clonal complexes, which represent sequence types with a similar central allelic profile based on MLST analysis. The significance of Listeria monocytogenes. So this is an opportunistic pathogen, and based on the most recent EFSA report, listeriosis is the 5th most commonly reported zoonosis in Europe. It has high overall EU case fatality, and this makes it one of the most serious foodborne diseases under EU surveillance. L. mono was the causative agent of 16 foodborne outbreaks, including 7 member states, 120 cases of illness, 83 hospitalizations, and 17 deaths. So listeriosis can manifest either as a bloodstream infection, meningitis, maternal neonatal infection, and the populations which are mostly at risk include the elderly, immunocompromised, and pregnant women. This illness is mostly associated with consumption of contaminated food, and this pathogen can have an increased virulence, which is related to the presence of Listeria pathogenicity islands. There's 4 of these detected. The first one is a PRFA-dependent gene cluster, and this is required for intracellular survival of Listeria monocytogenes. LIPI2 has only recently been discovered in L. mono isolates, and LIPI3 and LIPI4 are more so associated with the most pathogenic isolates Listeria, because these are involved in survival in neutrophils, and they also intensify central nervous system and maternal neonatal invasion. So the life cycle of Listeria through the food chain, it can enter the food chain kind of at any stage, from animals to the farm environment. Vegetables can become contaminated from soil or the use of manure as fertilizer. And then these contaminated products can enter the food industry, where isolates of Listeria can persist for very long times and cross-contaminate within that environment. And a lot of the time, products which are contaminated with Listeria are ready-to-eat products, which don't require any further processing before consumption, which ultimately then leads to human disease. So the persistence of Listeria monocytogenes in food production. So this is a really ubiquitous organism which is really ubiquitous in nature and has the ability to survive a range of stressful conditions. And its persistence has been linked to stress resistance, such as low pH and high salt concentrations, bioinflammation, and also increased tolerance to disinfecting agents. This results in difficulty managing and controlling this pathogen, which results then in a major concern for the food industry and public health. There are varying definitions of persistence in literature. Within our study, we define this as highly related isolates detected at least 3 times with a minimum of 1 year between the first and the last isolation. So moving on to my own study, I had 276 Listeria monocytogenes isolates, and these were collected between over a 7-year period between 2014 and 2020. The sources of these isolates included raw and cooked meat and environmental samples. And these were all sequenced on the Illumina MySeq. So the sequences were processed, and then I used a variety of different bioinformatic tools to try and characterize my isolates, as well as extract some specific information that I wanted to know about them. So I used 4 typing methods, and each one being more discriminatory than the previous. So I performed some serotype in silico methods, MLST in silico, CGMLST, and at the end, SNP analysis. I also assessed the collection for presence of virulence markers, biocide and stress resistance genes, and also some mobile genetic elements. So, as I said previously, the majority of clinical cases are caused by serotypes 1, 2A, 1, 2B, and 4B, and 41% of isolates within my collection were of these serotypes. 1, 2A is most frequently isolated from food, and 34% of my isolates accounted for this. 4B is implicated in the majority of human epidemics, and 2% of my isolates were of serotype 4B. 1, 2A and 1, 2C typically carry a premature stop codon mutation in the internal A gene, which is involved in the invasion of Listeria, and this renders it hypovirulent. Majority of the samples within my collection belong to serotypes 1, 2A and 1, 2C, and this was 93%. So moving on from that, I performed MLST in silico, and this is where I was able to assign clonal complexes to my isolates based on a 7-gene scheme. And the majority of these isolates belong to lineage 2, the lineage 2, because this made sense because they're more so associated with food production environments. And isolates belonging to clonal complex 8 and clonal complex 9 were found among all 3 sources within this study, so the raw meat, cooked meat, and environmental samples. Isolates belonging to clonal complex 8, 9, and 121 were presumptively persisting within the food production environment because, as I said, they were detected at least 3 times with a minimum of 1 year between the first and the last isolation. So further on from this, I was able to perform some CGMLST, and this is more discriminatory than just standard MLST in silico. So this assigns clonal types, CTs, and they are assigned according to 1,748 gene scheme, and these can differ by up to 7 allelic mismatches, so they're very highly related. Sublineages are also assigned based on the same scheme, however, these can differ by up to 150 allelic mismatches, and these were concordant with the clonal complex assignments from the MLST scheme. So this scheme comprises really highly conserved core loci, which represents 62% of coding regions from the L-mono EGDE reference strain. And I had 27 CTs detected among my collection. And the persistent CTs were identified within the presumptive persistent clonal complexes, which included 2 CTs in clonal complex 8, 3 in clonal complex 9, and 1 in clonal complex 121. So lastly, to characterize isolates, I performed SNP analysis, which is the most discriminatory characterization method of all of the 4 that I had applied. And this identifies SNP differences between highly related isolates. So within those persistent CTs, I found that my isolates only had between 0 to 59 SNP differences, which meant they were very highly related. And once I was able to characterize them, I then moved on to find out any other specific information I wanted to know. So their virulence profiles or kind of reasons as to why they could be persisting within food production. So first of all, virulence, the genes of interest were the lippies, which I spoke about earlier, as well as internal A and B and AUT, which are all involved in invasion. So the internalins were present in 100% of the isolates, and genes belonging to Lippy1 were present among all isolates, except ACT-A had a premature subcodon mutation among 8%. This is involved in intracellular mobility, and mutations in this cause unknown virulence. So we can't say for sure whether they're hypo, intermediate, or hypervirulent, even if they do have the additional Listeria pathogenicity islands. AUT was detected in 67% of the isolates, and LIPI3, which is involved in survival in neutrophils, was detected among isolates belonging to clonal complex 288, 217, and 1. And these are lineage 1 isolates, which are most so associated with listeriosis. Moving on then to biocide resistance and stress survival islands. So benzalkonium chloride is a biocide really relevant to listeria. It's commonly used in food production, and there were genes which render them resistant to benzalkonium chloride present in the collection. So the first one was the MDRL-YFMO. This is a multi-drug efflux pump known to be involved in benzalkonium chloride resistance, and this was present among 100% of the isolates. BCR-ABC, which is a cassette involved in benzalkonium chloride resistance, was present among 20% of the collection, and some other genes detected were causing resistance to acridine orange and ethylene bromide, which is LDE, that was present in 100% of the isolates, resistance to copper among 71% of the isolates, hydrogen peroxide among 20% of the isolates. Then moving on to the listeria stress survival islands, stress survival island 1 was present among 92% of the isolates, including the presumptive persistent CC8 and CC9 isolates. And stress survival island 2, which is known to be only detected in clonal complex 121, was detected in all of the CC121 isolates in this study. Moving on to some mobile genetic elements, I had detected intact prophages among 22% of isolates in my collection, which included 48% of isolates within the presumptive persistent CC8 and 4% of isolates within CC9, and questionable and incomplete prophages were disregarded as they lacked many of the structural phage genes. Of the isolates harbouring a phage insertion, the majority, which was 72%, were within the COMK gene, and COMK associated prophages were detected among 5 of the 6 persistent CT types. And a prophage insertion within the COMK gene has been linked to virulence, biofilm formation, and persistence in food production facilities. So some concluding remarks. So in food processing businesses, surveillance and early detection are crucial to control the occurrence and cross-contamination of Listeria monocytogenes. And in this study, whole genome sequencing-based approaches accompanied with bioinformatic analysis demonstrated to be a useful surveillance tool and facilitated the detection of presumptive persistent isolates within the food production environment over a 7-year period. And then these findings can be used to improve management of the food processing environment, especially sanitation plan adjustments. Some of my further work within my PhD then will focus on detection of ecophysiological genes related to stress resistance and biofilm formation, and also I will be working on some evolutionary experiments to assess effects of sub-inhibitory concentrations of biocides on Listeria monocytogenes, because that's more of a real-life representation of what goes on in food production. So that's it for me, and thank you all for listening.
Thanks, Natalia. Do you have any questions from the audience? Yes.
My question targets the plasmids. So did you have a look whether they harbor any plasmids?
I have had a look. I didn't find anything that was really pushing me towards identifying them as persistent or non-persistent isolates. So I actually hadn't included the results of this in this presentation, but I will be having a closer look at that just to see how that correlates with everything else. I think we just really wanted to focus on the phages for now, but yeah, I will definitely have a further look at that as well.
Thank you.
Thanks. Any more questions? One.
Hi, thanks for a great presentation. For the evolutionary experiment, how are you going to be doing that? Is it just going to be kind of mutation generating, or?
Yeah, so basically I've started that already. So what I'm doing is trying to grow biofilms on glass beads, and I expose them to either 0.25 or 0.5 of an MIC, and then reculture them every 3 days, and make sure to keep each of the passages, and then I will look at a midpoint and an endpoint to see if there's any mutations or any phenotypic changes.
Cool, thank you.
All right, last question. Here.
Yes, just a question. So the isolates were from 2014 to 2020, and you had 0 to 59 allele differences. So how would you estimate relationship between the isolates, given the fact that it's over a time frame over 6 years? Can you say something about that?
Yeah, that's the thing. So, certain of the CTs had 0 SNP differences, and they could have been detected over a few years, whereas certain ones had up to 59 SNP differences, which still isn't a huge difference, but you would expect them to mutate a little bit over time. So even though they have up to the 59 SNP differences, you'd still be able to say they're quite highly related. But obviously over a 7-year period, you would expect certain SNP differences.
Thank you.
All right. Thanks, Antelia, for
Thank
the
you.
excellent presentation. All right, next presentation, you have to bear with me. I'm from Alberta, Canada, where we only have half a year of winter, so the climate is quite different from Athens. The talk today is a small part of a larger project. Just a bit of background on the beef production in Canada. The beef industry in Canada is very consolidated, which means that you only have a few large plants, which processes a large number of cattle every day. And that also, we have a zero tolerance of E. coli O157H7. So those two factors pretty much drive the implementation of multiple antimicrobial interventions. For example, pasteurization of carcasses or organic spray and so on and so forth. And we have seen improvement in the safety of meat reflected by the reduction of incidence in human infections with O157. However, there are also concerns over whether those treatments would lead to increased resistance of bacteria in the meat processing environment, which will go to the greater environment. So the larger project is about to assess along the meat production chain how the resistance of E. coli change from cattle to the end product. So that's a bit of background on the project. Today I'm presenting a part of that project, genomic analysis of biofilm-forming and non-biofilm-forming E. coli populations from meat processing environment. Just a bit of background on E. coli. We all know E. coli is a commensal of warm-blooded animals. And they can, in addition to their primary host, they can also survive in a variety of environments. And E. coli can be divided into 8 phylogroups. And there are some relationship between their phylogrouping and their ecological niches. And even though most of E. coli are harmless and some can be beneficial to humans, but there are a fraction of E. coli that can cause disease. For example, Shiga toxin producing E. coli, which has a very low infectious dose. Even though the concern of O157 on meat, particular beef, is huge, but they don't exist in high numbers on meat products in commercial settings. So generic E. coli is often used as an indicator in food processing, including meat. For those of you who don't work with the meat, primary production of meat, it can be categorized into 3 primary stages. One is from the live animal to dress the carcasses. 2 is the chilling that would chill the warm carcasses to a low temperature to prevent bacterial growth. And then the chilled carcasses are fabricated into cuts, trimmings, and whatnot. So that's the general production process of beef. And as I mentioned, generic coli is often used as hygienic indicator where you would sample and test the chilled carcasses and the final products. The number of generic coli has dropped quite significantly from the 90s to early 2010-ish. For carcasses, there's about 1,000-fold improvement or a decrease in terms of the numbers. And the same trend, but to a lesser degree on cuts and trimmings. And you'll see the numbers on cuts and trimmings are often higher than the numbers of E. coli on chilled carcasses. Before the genotyping era, there were two theories. One is that it's from incoming carcasses because you don't know which strain they are. With the genotyping, we figured out that some of the E. coli on equipment actually are of the same type as the E. coli on final products, which shows equipment can be a significant source of contamination of E. coli on final products. We did some work on evaluating the response of E. coli that are persisted in meat plant and that are transient, their response to sanitizers, to antimicrobial treatment. We did not find much difference between the E. coli that are supposed to be naive and E. coli that are supposed to be after the meat processing that have presumably that have been exposed to those treatment, antimicrobial, biocides, and whatnot. And however, those E. coli, the persisting ones do have a stronger biofilm forming potential compared to their transient counterparts. So what are biofilms? Biofilms are surface-attached microbial communities embedded in self-produced extracellular substances. Because of those physical, chemical projection of the shelter and the physiological state of the cells in biofilms, they are more resistant to chemical treatment. For biofilm forming potential, two phenotypes are often regarded as indicators for strong biofilm forming potential. One is cellulose production, and the other is curly production. We did some work to look at the biofilm forming potential of E. coli from cattle, from meat, from equipment before after cleaning. What we noted was that the proportion of E. coli that forms strong biofilms increased. With the isolates from equipment had much higher fraction of biofilm formers compared to cattle, compared to meat, and in the middle of the chain as well. So that takes us to the objective of this smaller study, that is to look at do those biofilm formers and non-biofilm formers differ genetically. The reason we did this was that we used the narrower environmental conditions because E. coli has open genome, is so diverse. We thought by using this, it could reduce some background noise. The approach we have taken is short-read whole genome sequencing combined with phenotypic assessment of curly and cellulose production. And the table shows a strain selection. Altogether, we included 114 strains, 113 meat and equipment isolates, one positive control. The selection rationale was that we had equal number of biofilm former and non-biofilm former from one source, except the last one, equipment after cleaning, where we evaluated 100 strains, we only found 3 strains that did not form biofilms under the condition we tested. So 114 strains. What we found, I mentioned earlier that there is some relationship between phylogrouping of E. coli and their ecological niches. For cattle source E. coli, they are mostly phylogroup A, phylogroup B1, you will see most of the groups would reflect that. However, there is a group that is equipment after cleaning, and they are primarily of phylogroup B1. Phylogroup B1 differs from other groups in terms of their environmental survivability. And this is a core genome tree. It's quite messy. What I wanted to draw your attention is that first I will explain the legend. The black font indicates biofilm formers. The blue are non-biofilm formers. The very inner ring is the source of collection. The middle ring is the phylogrouping. What I want to show you was that you will see close clustering of very close strains. The non-biofilm formers, the biofilm formers, and a mixed biofilm former, for example, clade 7, and mixed biofilm former, non-biofilm former, clade 1. So the biofilm forming strains are spread on the tree, and there are clusters of mixed in terms of biofilm forming. Next, we did some clustering by principal component analysis. We did not see much separation of the biofilm forming and non-biofilm forming E. coli. We did a more detailed analysis using Scurry and PySeer. Those two apps are to analyze genome-wide association between traits and gene content. We're using the definition. We're using criteria of 60% as specificity and sensitivity. We did not find any gene that is overrepresented in the biofilm formers. And this is a phenotypic assessment. What I want to show you, one is that there is, for the biofilm, for the cellulose positive and curly positive, there is a stronger biofilm forming potential compared to the negative group, the trait negative group. However, you see for the non-cellulose producers, non-curly formers, you also see biofilm formers, and vice versa. You see the trait positive cellulose producers or curly producers, they didn't form biofilms. And we look at the genes that encode cellulose production and curly production. Want to explain the legend a little bit. The vertical line separates the left cellulose production genes, the right curly production genes, the horizontal line, top trait negative, bottom trait positive. So and the color, the blue to red, indicates the presence and similarity of a particular gene to the positive control. And the gray color indicates absence. What I wanted to point out is you look at the cellulose positive trait, you will see isolates with missing one or more genes in the cellulose production, here, 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 here. Of course, there are isolates that are fully equipped with those genes, but are cellulose negative. For curly, all curly producers have all the genes, but they're curly negative ones that did not have, they're curly negative ones that have all the genes present as well. So, in summary, the E. coli group recovered from equipment after cleaning differed from others in their phylogrouping, and there was no clear delineation between biofilm forming and non-biofilm forming E. coli group by core genome, gene content, or GWAS analysis. And the curly and cellulose expression are positively correlated with biofilm formation on a population level, but not always on individual strain level. I think this takes us to the question, one of the major reasons we study biofilms is to get rid of them. And we often use a strain-targeted approach to see this strain produces biofilms, and we target how to get rid of it. My thought on this is that it might be time that we step back, we could treat them all as biofilm formers, and then devise interventions to remove them. And that is end my talk, If you have any questions, I don't know the time. I think we're good with time. Any questions? Oh, there's one.
Thank you for your nice talk.
Thank you.
I have a question. Which method did you use to characterize your biofilm formation? And which criteria do you use to say it's non-biofilm former or biofilm former? And I have another question about the temperature. Which temperature did you choose to be realistic in terms of meat production?
Excellent questions. Question number one, I should have mentioned the biofilm forming. We assessed the method we used was crystal violet staining, so to stain the total biomass. The other question, the temperature, we use 15 degrees centigrade. We know E. coli don't grow. Most of E. coli don't grow under the temperature of 8. For meat operations, the fabrication floor, the temperature is about 8 maximum. Sometimes it can be as high as 10. However, during downtime, the temperature can go up to 15. And also when the equipment is running and there's friction, and the temperature could be higher than the ambient temperature. So we use 15 to simulate a realistic scenario. So I guess I'm done. Now we introduce the next speaker. All right, our last but definitely not the least speaker is Lu Jiang. Her presentation is Detangling the Complex Pathways Between Dietary Molecular Composition, Gut Microbiome, Enzymatic Biotransformation, and Health Outcomes. Ms. Jiang.
And good evening, everyone. I'm Lu Zhang. I'm a PhD student studying in Leibniz Institute, Hans Knoll Institute. And first, I want to thank the organizer for providing me the opportunity to present my PhD project. And my talk today entitled Detangling the Complex Pathways Between Dietary Molecular Composition, Gut Microbiome, Enzymatic Biotransformation, and Health Outcomes. First, I will start with the background interaction. Non-communicable disease, namely NCD, is one of the leading causes and disability that occurred all over the world, affecting population in developed countries as well as in developing countries. An estimated 36 million deaths, 63% of the 57 million deaths that occurred globally are due to NCD. The major NCD, such as cardiovascular disease, cancer, diabetes, they all shared 4 behavioral risk factors, and one of them is unhealthy diet. Diet is so vital to human health, and indeed, the rise in NCD has a causal link to the global dietary pattern that are increasingly becoming westernized, being characterized by high levels of sugar, salt, fat, and lack of fresh fruits and vegetables. Some studies have been done in this field. Studies have shown the increase of whole grain intake can reduce the risk of cardiovascular disease, and omega-3 fatty acids have potential health beneficial in preventing cognitive decline. And DASH diet works more efficiently than the routine care in adolescents with hypertension. As shown in many dietary intervention studies, diet can induce the alteration in gut microbiome, and emerging evidence also showing that the dietary response is personal-specific, and some particular bacteria species can be predictor for the response of the diet. On the right part of the slide is a study from an Israel cohort that they measured the postprandial glycemic responses in the population based of 800 participants. That they demonstrated that PPGR is highly variable in individuals, And they developed an algorithm not only including clinical parameters, but also microbiome features to accurately predict the PPGRs to the real meal. They suggested that rather than the genetic makeups, non-genetic factors, including microbiomes, plays an essential role in personal response to the diet. Currently, the microbiome and dietary research mainly focused on nutrients and food categories. However, more than 26,000 distinct small molecules exist in our food. And plants have particularly rich chemical composition. A few studies between the microbiome and molecules in plant-based food has been elucidated. For example, gut microbiota metabolize neurotransmitters and bioactive compounds from polyphenols which are enriched in tea. However, analysis of plant diet at small molecule level is still lacking and needed. Thus, based on this background, we integrate dietary phytochemicals with microbiome functional traits. Here we reference the microbiome enzymatic function by using 11 databases. The dietary phytochemicals is from our in-house molecular level database called NutriChem. That in this database, it contains 1,800 plant-based food and recorded almost 8,000 phytochemicals. So in total, the NutriChem database contains almost 20,000 pairs of plant-based food and phytochemicals. In addition to collected the enzymes and phytochemical interactions, we also collected 10 other biochemical reaction databases to build this link. And then we combine these links with our collected public cohort data. That this data we collected are more than 3,000 shotgun metagenomic fecal samples, and they are mainly from 5 different continents. So in total, we obtained more than 8,000 microbiome enzymes and phytochemical pairs. And based on these links and our collected public cohort, the first question we wanted to explore is how many % of the microbiome enzymes involved in the metabolism of diet phytochemical molecules, and how many the phytochemical molecules these microbiome enzymes are related to. As for each sample, we found that on average, 73.8% of the microbiome enzymes that participate in this dietary processing procedure, which indicates that majority of the microbiome enzymes participate the dietary-based food processing procedure. And then on a population level, we find that these enzymes with EC number that can link to at least 1 phytochemicals that in total linked almost 1,000 diet phytochemical molecules, with a range from 670 to 700 in each continent as the average number. And in total, it linked more than 1,000 plant-based food with an average from each continent from 1,000 to 1,017. And surprisingly, we found that microbiome enzymes from different continents, they are processing very similar dietary phytochemical molecules and very similar plant-based food. We found that 750 are shared among continents in terms of dietary phytochemical molecules, and more than 1,000 are shared among continents in terms of plant-based food. And then next, we were curious whether this percentage of the diet-related enzymes they varies with the overall microbiome enzyme and species diversity. So in this case, you could see the upper panel that we did a correlation between the enzyme diversity with the diet-related enzyme percentage. In the lower panel that we did this correlation between the species diversity and the percentage. And we used Spearman correlation as well as linear mixed model to control for the study differences. And the result demonstrated that you can say there is always a significantly negative correlation with a p-value less than 0.05 between this percentage in the species diversity and enzyme diversity, whereby we hypothesize that there exist a cold and stable microbiome groups and enzymes that are responsible for the dietary processing, and they don't increase with the increasing gut floral diversity. And then we also compared the diet-related enzymes with the non-diet-related enzymes. The way how we define them is based on whether this enzyme with EC number can link to at least one phytochemical molecules. If it can link to phytochemicals, then we define it as dietary-related enzymes. If not, we define it as non-dietary-related enzymes based on our current database. So first, we compare the two profiles in terms of the diversity. And you could say here on an enzyme by enzyme basis, there is a significantly higher diversity in dietary enzymes than the non-dietary enzymes. Except this, we also compare the abundance between these two enzyme profiles. And we find that on a sample-by-sample basis, which means that for each sample, we can group the enzymes into diet-related and irrelevant ones. And then we calculate the median value of these enzymes. And then we used a beer constant rank test to compare these two profiles. And you could see here also, on the left part of the slide, there is a higher significantly abundance of the diet-related enzymes than the irrelevant ones. And we observed, you can see on the red part of the slide, that we observed a very consistent result if we compare the two profiles within the continent. So the result demonstrated that there's more diversity and more abundance of the dietary-related enzymes, which means that even though one species encode dietary enzymes cannot work, there is going to be some backup species that they can encode these dietary-related enzymes and makes the diet function works well. So far, we have explored the molecular level and also the enzyme level. So next, we dived into the species that are responsible for the processing of the plant-based diet procedure. So as for each, on average of 39 species per individual is sufficient to cover almost all enzymes involved in the metabolism of small molecule in plant-based food. That here we'll use the gradient algorithm. And then we will also define this species list as minimum species list. And then we did a correlation between the species prevalence and the species abundance with this minimum species list times. And we find that there is a significant correlation between these two. So it demonstrated that the more abundant and more prevalent the species is, the more likely this species exists in this 39 species list. And we also extend the level to the compound level as well as the whole population. So in this case, it means that if we want to cover 50% of the all dietary compound for the whole population, we will need on average 44 species, and in 100 times subsampling the dietary compound. And if we want to cover 100% of the compound, we'll need 58 species. And if we look into the details of these 58 species, surprisingly we find actually 8 species, they have linked so many phytochemicals, and these 8 species can be enough to cover 80% of the compounds with a big function in a big potential in application in the future. So to summarize, in this research, we gained insight between the microbiome, enzymes, and phytochemicals by using 11 databases in a healthy cohort with more than 3,000 shotgun metagenomic samples. And the result demonstrated that on average, 73.8% of the microbiome enzymes participate in the diet processing function. And we also found that this percentage negatively correlate with the enzyme diversity and as well as the species diversity. The result demonstrated that there's high abundant and high diversity of the drought-related enzymes than the irrelevant ones. And also, we find that on average, 39 species per sample is enough to covering 90% of the drought-related enzyme function. And 58 species will be enough to cover 90% of the drought-related compounds. And impressively, 8 of the 58 species can be responsible for precisely 80% of the compound. Finally, I would like to thank my supervisor, Professor Yanni Panigiato, and my colleague, Dr. Ni, for providing me a lot of guidance on the project. My collaborator, Andrea, that we are doing this project together. And also Dr. Chen for providing some calculation in the species level. And I also want to thank the team for the great scientific atmosphere and the funding for providing me the chance to start my PhD project. With that, thank you for listening, and I'm happy to take questions.
Thank you, Ms. Zhang, for the excellent presentation. Do we have any questions? No? If not, thanks again for the presentation. And thank you all for attending, and enjoy the rest of the conference.